my name is Elliott Cole, and I'm going to be briefly describing the DNA binding protein NotI. So NotI is a type 2p restriction endonuclease, which binds to and recognizes an 8 base pair DNA sequence, then cleaves specific phosphodiester bonds in the DNA backbone, generating 4 base pair overhangs. It is a homodimer, but throughout the presentation I've removed one of the monomers for image clarity. Now, the remaining slides contain images of NotI, which I've generated in Chimera to highlight the following key folds: the catalytic domain, the recognition helix, the metal binding domain, and the clamp domain. And the story really starts with the metal binding domain, which contains a cis-4 motif that positions sulfur atoms in a tetrahedron around a bound iron ion. And interestingly, mutations to 3 of these cysteine residues, namely 42, 55, or 81, result in enzymatic inactivation, despite the fact that the iron ion is located 15 angstroms away from the active site and isn't suspected to be involved in catalysis. So this fold plays some sort of structural role, and indeed it's proposed that the metal binding domain positions the clamp domain in such a way where it can form this network of stabilizing hydrogen bonds with the DNA. That's a great example of allostery in action. Now here's an example of one of those hydrogen bonds. I picked this one out because it interestingly occurs between the protein backbone and the DNA nucleotide base. So we'd expect it to be conserved for any other amino acid substituted in this position, provided that the new sequence adopts the same protein fold. And the rest of the base pair recognition comes from the recognition helix, which sits in the major groove of the DNA and interacts directly with many nucleotide bases. Now together, both monomers fully saturate the central 6 base pairs of the recognition sequence with hydrogen bonds. And although there are other forces at play in DNA binding, these hydrogen bonds are very important because they will only all form in the case where NotI is bound to its target DNA sequence, but won't form when non-target sequences are bound. And this is what produces the effect of sequence specificity, because these hydrogen bonds lower the binding energy and make NotI more likely to be found bound to its target sequence than its non-target sequence. And that's a well-established consequence of statistical mechanics and the Boltzmann factor, which is shown down below. Now, to contrast the last interaction, this example involves hydrogen bonding between a DNA nucleotide base and the amino acid side chain, and so it's explicitly specific to the protein sequence. Now finally, there's the catalytic domain, which consists of the central alpha helix surrounded by a mixed beta sheet, and the active site of this catalytic domain contains 2 calcium ions, which are shown in pink, and the arrangement fulfills the canonical PD-DRE-XK catalytic motif, with slight variation, substituting proline with phenylalanine and lysine with glutamine. So that's all I have. This first paper was my primary reference, and thank you for your consideration.